So good morning everyone. Good morning everyone. Welcome to the lecture. Welcome to the series of immunology, and today we are talking about another part of this particular section, that is molecular structure and types of immunoglobulins. So in previous lecture what we did. See, in previous lecture what we did is very simple part. Today we are talking about the molecular structures in immunology, and in previous lectures what we did. In previous lecture, we had a very clear-cut understanding regarding your general properties of immunoglobulins. What are immunoglobulins? How you will be defining those immunoglobulins, and what is the difference actually? But it's a minor difference between immunoglobulin and antibody. Then we had established a basic understanding regarding different classes of immunoglobulins, and we had discussed two specific classes in very depth. That is IgG and all its subtypes with all the properties, saying that it's the maximum, it's the highest concentration of IgG that is present inside the serum. Then we had talked about IgM. So today's lecture will be focused on further describing the immunoglobulin types, and we shall be able to discuss immunoglobulin A, E, and D in depth today. So by the end of the lecture, I hope that you will be having a clear-cut understanding of the complete immunoglobulin family and the 5 classes of the immunoglobulins that are present inside our immune system. So just to recapitulate, just to bring back the previous lectures memory, what we had discussed was very simple. What we had discussed is this particular slide. In nutshell, if I say this particular slide, where we had talked about there are 4 chains constituting the immunoglobulins. 2 are heavy and 2 are light. And then we have 4 domains in heavy chain, 4 domains in heavy chain, and 2 domains in light chain. And the upper domain in both heavy and light chain is actually the antigen binding region, which is, I am circling here, and this is called as variable region. This is called as variable region, whereas the other 3 are constant region. Other 3 are constant regions. So if this is the antigen binding site, then this particular portion is responsible for activating the immune cells. What type of activation am I talking about? See, there are many receptors present on different cells. If we are talking about B cells, B cell contains a receptor called as FC receptor, which is responsible binding to this particular segment of constant region. Similar case is with the macrophages. They recognize this particular part of the antibody or the immunoglobulin in order to identify where the antigen antibody complex is present. And if the antibody has to cross the placenta and has to go from mother to the fetus, that particular transfer of this immunoglobulin from placenta is mediated by this constant region, that is the constant region number 3. So these all functions are what we have deliberated in the previous lecture, and we had also discussed there are 5 types of classes. Among them, 2 we have discussed in detail. Today we are focusing on alpha, delta, and epsilon, or called as A, D, and E. And in yesterday's lecture also, just to tell you about the IgM, that we had established that there is a J chain which is holding 5 immunoglobulins of IgM, making it a pentamer. So it's a pentavalent. How it is pentavalent? It can bind to 5 different sites of an antigen, 5 different antigens. Not 5 different. It can bind to 5 antigens of the same specificity. See, if this is specific towards a single type of antigen, all the others will also be specific for that particular antigen. So it's a pentavalent. It has 5 valencies. And if you go for its sedimentation part, then the sedimentation rate and the Sidberg unit is coming around 19S. So always remember IgG 7S, IgM 19S, and that is what we had discussed yesterday, and we had established its functions also. So let's go ahead and talk about another immunoglobulin, that is IgA. Now, we had a very clear-cut understanding that inside our serum, the highest concentration of immunoglobulin is IgG, right? Third highest is IgM, and second one is IgA. So you will see that it constitutes about 10 to 15%, 10 to 15% of the total serum immunoglobulin. So immunoglobulin G was around 70%, 70-75%, and there is 10 to 15% of IgG. Now, the unique part of IgG is what? It exists in both monomeric as well as dimeric form. It exists in both monomeric as well as dimeric form. What is monomeric and dimeric form? Let us see here. Let us look at this particular image. Let us look at this particular image and then we'll discuss. So you see a single structure. A monomeric structure is in this manner, which is constant for all the immunoglobulins. This is a monomeric structure. Another structure of the same type came, and a linker between them is present. Another structure of same type. If this is specific for this antigen, this antibody, another will also be specific for this antigen. So both the antibodies are connected by a molecule called as J chain, like it was present in IgM. Now the J chain is linking the constant region number 3 of both the immunoglobulins. So the tail piece is now linked by J chain. It is present in this fashion. Now it's because there are two components, because there are two components of immunoglobulin A, so it is bivalent. We call it as bivalent. We were calling IgG as monovalent. We called IgM as pentavalent. So this particular IgA is nothing but IgA is bivalent. Why bivalent? Because of two units. Because of 2 units. Okay, fine. Then there is another part that is highlighted in yellow in this particular image. This particular part is called as secretory component. This particular part is called as secretory component, and this particular proteinaceous part, which is present in the dimeric form, which is present along with the dimeric form, is actually responsible for secretion of this immunoglobulin outside the cell. So you see what properties are there. It's the second highest property. It's the secretory immunoglobulin. If somebody asks you what among all the 5 types of immunoglobulin, which one is secreted out? See, you have to clear cut understand. You will say that every immunoglobulin can come outside the body and can perform its function outside the cell and can perform its function inside the serum. But initially, suppose this is a B cell. Immunoglobulins are secreted from B cells, right? Plasma B cells. So initially, all the antibodies or immunoglobulins are bound on the surface. As soon as they are coming under the exposure of an antigen, then only they are released from the membrane. They are released from the membrane. But this immunoglobulin, the IgA, is always secreted out. It is not membrane bound. It is taken up inside the cell and then it is released or secreted out. And the secretion is the property of that secretory component. So in lab, suppose you designed this particular antibody structure, you connected it with the J chain, fine, and you never added secretory part. So you will see that it is taken up by the cell, but it is remaining inside the cell. It is never coming out. So that secretory region or secretory component that we are adding is responsible for its supply or secretion outside the cell. And that is the reason that this particular IgA is present in secretions of your body, like tears, like saliva, gastric secretion, pulmonary secretion. So all the secretions of the cells which are present, they are actually responsible. This antibody or this immunoglobulin is actually responsible for fighting in those secretions. So how that particular part is achieved, that will come to in that part. Now another part that we should remember here, when we were talking about IgG, this is IgG what I am making, we said that this particular portion that I am highlighting, this portion was responsible for activating complement system, for activating the macrophages, right? For activating the macrophages. Now, you see this IgA structure, and you will see that this particular region is not free to interact with these cells. That is the reason why it is not able to activate the complement pathway. It is not able to activate the complement pathway under normal condition. When it gets aggregated, and then only it might activate. Fine. So in the serum, when we're talking about IgA, is always in monomeric form. But when we are talking about the secretory part, its secretion outside the cell, then it goes into dimeric form, which is joined by a J chain. Now, this secretory component is actually responsible for its transition from the cell. Now, the question comes, when B cells are preparing it, when B cells are preparing it, they are releasing it in the serum. They are releasing it in the serum. And suppose I am talking about pulmonary secretions, right? Pulmonary cavity. There are epithelial cells that are present in my lungs, epithelial cells. How these epithelial cells are taking this particular immunoglobulin inside and then secreting out in terms of secretion. See, my tears, they are coming from my eye, from lacrimal gland and the cells which are present in my lining of my eye. So my eye is not filled with B cells, right? So how this particular structure manages to enter inside the epithelial cells, that we should talk about. So that is the responsibility of this particular secretory component. Now this particular secretory component, uh, which is uh, which is actually responsible for the secretion, that as I said earlier, this is taken up, or this is derived from receptors that are present on these cells. Let me show you in just a sec. Let me show you in brief. Let us focus on this particular thing first. So what is happening? See, initially when the dimeric form is formed by B cell, it is not containing any secretory component. So B cell has released this particular dimeric form inside the dimeric form. It is taken up by a receptor on the epithelial cells. This receptor will recognize this particular dimeric form, and then based on this receptor binding, this complete complex will be endocytosed. It will be taken back inside the cell, inside a vacuole. Once it is back inside the vacuole, the receptor is present in two forms, in two parts. If you see, there is a stalk which is binding to the membrane of the vacuole, and there is upper part which is specific for the dimeric immunoglobulin A. So what happens next? It releases the molecule of dimeric immunoglobulin A bound with J chain along with the portion of the receptor which was present on the epithelial cell. And this particular portion is actually the secretory component of immunoglobulin A. So you see, B cells are not making the secretory component. The secretory component is actually derived from the surface receptor which is present on the cells of these linings with or the epithelial cells. So the portion of this particular receptor is actually responsible for its secretory part, and that's why we say that it is derived uh, from the poly Ig receptor present on the serosal surfaces. So that is the mechanism actually how this immunoglobulin A is secreted out, how it is formed, how it is retained by the cells, and then attached with the secretory component. So let's talk about in brief the function of this secretory immunoglobulin A compared to the other part. It is responsible for providing the immunity in the mucosal part. In the mucosal part or in the localized region, we are not talking about the systemic immunity. No, we are talking about the localized part. So in tears, if IgA is present, it is providing the immunity in that area only. If in pulmonary section, that particular secretory form is present, that is providing the immunity in that section. So wherever the secretion or the secretory fluid is there, based on there, then it will be providing the immunity in that particular section, and that comes as mucosal part. It's the mucus or the mucosal immunity that is majorly contributed by IgA. So based on that, we can say that it is effective against what? It is effective against the viruses like polio virus or the influenza virus, which is affecting the respiratory tract, and it is fighting against the fluid, the pulmonary fluid or the mucosal part of our respiratory tract, which is rich with immunoglobulin A. Or we can say that the secretion which is coming out as a breast milk, it is very rich in IgA. So when the question comes that which immunoglobulin is present in colostrum, or which immunoglobulin provides the protection to the baby via feeding, that part is immunoglobulin A. That part is immunoglobulin A, and it provides very, very fine protection to the intestine of a newborn baby. That is why doctor recommends that yes, breastfeeding should be done. So the milk is very rich in IgA. It reaches to the intestine of the newborn baby, and there it fights with the microbial flora, which may cause the impairment in the normal development of the intestine. And apart from it, when we're talking about the dimeric secretory immunoglobulin A, of course, I told you it is synthesized by plasma B cells, the dimeric part, and then it is released and taken up by the epithelial cell. And these epithelial cells are responsible for providing the secretory component. Now, this is actually the image, the real-time image that you should be able to understand. See, this is your plasma B cell. This is your B cell, the plasma B cell. Fine. The B cells are circulating inside the blood. So blood is reaching to every cell type. Now it has reached to epithelial cells. The B cells has reached to epithelial cell. This B cell will release the dimeric form. It will release the dimeric form of IgA. Now this IgA dimeric form will be recognized by the specific receptors which are present on these epithelial cells, and these receptors are called as poly-Ig receptor. These are called as polyimmunoglobulin A receptors. Now they will bind to this particular antibody, and you will be very surprised to see that in this structure that I am talking about, these receptor binds to this region. The receptor binds to this region. Now when they bind, they go for endocytosis, they form a vacuole. Normal endocytosis process is there. This endocytosis, after binding to the receptor, is called as receptor mediated endocytosis. Now, once the endocytosis is done, then enzyme inside this vacuole will be activated and it will cleave off. It will cleave off the receptor, and then the epithelial cells will be further secreting the active secretory immunoglobulin A inside the lumen. So what is happening? What is happening? Here is the systemic regulation of blood. Here is the barrier of epithelial cell, and how the immunoglobulin A is managing to cross this barrier and come inside the lumen as part of the secretory activity. Now it is very active and is ready to fight against any infection. And that's what we have discussed when I was telling you regarding the secretion part of this particular immunoglobulin A. So that is all about the function of immunoglobulin A. Why we are discussing in depth? Because it's the second most abundant immunoglobulin inside our system, inside our immune system. So depending upon the we had already discussed there are four different types of immunoglobulin G based on the sequences or the amino acid sequence of constant region of heavy chain. Similarly, based on the amino acid sequence of constant region of heavy chain, IgA is of two types, IgA1 and IgA2. So there are two subclasses that we are talking about of IgA. And that is all that you should remember regarding that particular IgA. Then let's talk about the fourth the immunoglobulin that we should talk about, that is IgD. Now, before going to IgD, I should tell you, if you will anytime if you isolate a plasma B cell, you will see two specific antibodies that are present in many number. This will be IgM and this will be IgD. So once the IgD is formed, it is expressed on the surface of the plasma B cell, and that expression on the surface can be taken as a marker, can be taken as an identifier if you want to identify the B cell among the mixture of that particular or among the mixture of the complete white blood cells. So let's talk about structure. It's very similar. It's very similar to immunoglobulin G. There's nothing, not much difference. I'll highlight the main difference part. There is a presence of extra tail piece, which is responsible for this particular anchorage that I was talking about. The anchorage that is there, this particular anchorage that I was talking about. This anchorage is possible because of an additional tail piece apart from the domains. Then next, in immunoglobulin, you see that only one carbohydrate moiety was present on constant region 2. But here there are multiple carbohydrate or oligosaccharides that are present on on the complete length of constant region. So there is a higher degree of glycosylation. There is a higher degree of glycosylation. But what is the critical part? If you see this structure and I divide in this part, the site which is responsible for binding to an antigen is never glycosylated, but the site which is responsible for activating the immune cells, that is highly glycosylated in this particular IgD, and that is the classical marker for identifying the IgD, whether it's IgG or IgD. So based on glycosylation pattern, we identify that yes, this is IgD actually. So it's a B cell surface that I told you. It's the fourth highest serum immunoglobulin inside our body, and it is not able to activate the complement pathway. The complement pathway is activated by IgG and in some cases IgA. So that is all about IgD. Then let's talk about IgE. The IgE part, it's again very important. Why? Because there is an extra domain like IgN. There is an extra domain like IgN, and we call it as constant region of E, epsilon actually, a fourth constant region of epsilon. So similar structures follow. There are four patterns, and you see the glycosylation pattern is now clearly demarcating how we can identify IgE. So it has the similar glycosylation pattern as IgG, IgD. Then it has crossed the variable, it has crossed the Heinz region, and it has managed to cause the glycosylation in the constant region first. But and there will be no glycosylation in the variable region. Why? Because variable region is the portion where the antigen will be coming and binding. So I cannot touch this portion. So immune system has established that this particular section should be static. It should be the same. It should be only its whole sole function should be binding to the antigen. So that is how we can differentiate. If I want to differentiate between the different antibodies based on glycosylation pattern, I can differentiate. Based on their length, I can differentiate. Based on the presence of the extra domain, I can differentiate. So all these properties based on our understanding on that structure are actually responsible for identifying different immunoglobulins in the laboratory settings or in the clinical settings where we want to identify what type of antibody or what type of immunoglobulin is actually present inside this particular serum. So if you talk about the properties, this is the least abundant immunoglobulin in the serum. So the order is very clear: G, A, M, D, E. That is the order of percentage of immunoglobulin inside the serum. It should always be remembered, right? Then what is the other property? See, this is very peculiar property. It is they do not require any kind of antigen binding. They are secreted in response to allergic reactions always. If you're talking about any allergic reaction, we had established that we'll be talking about allergic reaction, then we'll be talking about the basophils, that we had established the understanding of basophils, the granulocyte, what we are talking about. So what it does, it goes and binds to the basophils or the mast cells which are producing the histamine. It will bind to the cells which are basophils or mast cells that are responsible for secretion of histamine. It will go and bind on their surface. Also, apart from allergic reaction, it is very potent fighter against any kind of helminth parasitic infection. We can talk about any kind of intestinal helminth infection. So this particular antibody will go and bind on the surface of that particular helminth, and then this particular region will be recognized by the eosinophils, and those eosinophils will come and release the granules on this parasite. So you see, it assists, it is providing a supporting part to basophils in terms of allergic reaction and to eosinophils in terms of fighting to the parasite. Because eosinophils, what is happening here? Eosinophils are responsible to degrade this particular element, to kill this particular parasite, but they cannot recognize it. So what is the connecting link? The IgE will be going and binding to the surface. These eosinophils will be recognizing the constant region of this particular IgE. They will be circulating in the blood, and then they will be coming to a static. They will be coming to a hold position once they have interacted with this particular constant region of IgG. And once the binding is achieved, then they will release the granules, which will be released on the surface of a live nematode, and these granules will cause perforations and digest this particular parasite. And this is how the parasitic infection is removed from our body. So that is the main function of immunoglobulin A. So it's a very low concentration that we have established. Its plasma half-life is the least, is the shortest plasma half-life. It's never present for longer duration inside the blood. And as per the daily production, what we are talking about from our immune cells is very low, and it's very sensitive to temperature. It's very sensitive to temperature. If the temperature reaches around 56 degrees Celsius, then it's completely inactivated in 1 hour. It's completely inactivated in 1 hour. Apart from antigen, see, please have a clear understanding. The immunoglobulin E will never bind to any antigen. Will never bind to any antigen. They are binding to the cells of basophils or mast cells. Also, they are binding to the parasites. They are binding to the parasites, specifically in the case of helminth. So on that notion also, they are binding to the cells of that surface. They are binding to the cells of the surface. So that is the function. And then when we are talking about the advanced function, then this IgE is actually the mediator of starting the hypersensitivity reactions that we'll be talking about in the later lectures, the type 1 hypersensitivity reactions, which is very, very important for our body to fight against multiple infections. So although it is present in very small amount, although it is secreted in very small amount, but it is the one which is actually responsible to fight against many allergens, many parasite infections, and the hypersensitivity reactions elicitation. So when we go for when we go to a doctor says that yes, let's go for the blood count and let's go for the antibody titer estimation. And if doctor sees that yes, IgE is abnormally increased, then it's a clear cut indication that either you have a very potent allergy, persistent allergy, or you are infected with a parasite infection in the form of helminth infection, and that's a clear-cut marker for this particular type. So after studying the complete scenario, let us summarize the complete part. Let us summarize the complete immunoglobulin part in a form of a table. So there are 5 immunoglobulins that we are talking about. In the structure part, IgG is monomer, IgA is both monomer and dimer, IgM is monomer and pentavalent, remaining 2 are again monomer. So the valency, when we are talking about, it is bivalent, it is tetravalent, it is again pentamer. So we are talking about 10 valencies, and then we are talking about again. Then other chains, J chains are present in IgA and IgM. The subclasses are present in IgG and IgA. The molecular weight is completely different, and that can be used. See, there is almost similar molecular weight of 150 in all the monomeric form. So if you want to identify differentiate these three, that it can be differentiated based on the glycosylation pattern. It can be differentiated based on the glycosylation pattern. Then the serum level is very clear. Highest is IgG, then A, then M, then D, and so you see the percentage of this particular. It's 0.003 mg, almost 0.3 micrograms per ml, 0.3 micrograms per ml. Half-life is highest for IgA, then it's for A. IgG is highest, then A, M, D, and E, and that is what we have established here. So if you see this region clearly, these are of very short half-life, but the immunoglobulin which will be present for longer duration of time inside your blood or inside your serum is IgG, and that is a reason it's the very, very important immunoglobulin in terms of fighting to the infection, and also the daily production is highest for IgG. Then if we talk about the intravascular tissue distribution, then intravascular distribution is highest for IgM and IgD. Why? Because it's been present at the surface of B cells. Then in terms of sediment coefficient, please remember it's pentavalent. That's why it is 19s. Otherwise, everything is 7s or 8s. Fine. Complement activation. This IgG is able to activate the complement pathway. IgM can also activate the complement pathway, but A, B, and E will never activate the complement pathway. It IgG can activate the phagocytosis. Phagocytic cells remaining will not be able to do so. Placental transfer only and only IgG except Ig2. All the three other isotypes will be responsible for transferring the immunity from mother to the fetus. So this is all about the summary that we are talking about. Like marker for B cells, only M and D are present because they are present on the surface. So this summary chart will help you in understanding in bringing the complete picture of all the 5 immunoglobulins to 2 slides where you can summarize the complete property of the immunoglobulins that we established in 2 lectures. So based on that, let's talk about the MCQs. IgG exists in, of course, monomeric form. The sedimentation coefficient of IgM, it was clear that is 19s. The classes of immunoglobulins have been recognized. There are 5 classes that we are talking about. I have not written here subclasses. I am talking about only the classes. Then bond by the heavy chain and light chain are associated together by what bond? It's the disulfide bond that we had discussed yesterday also. Then pentameric form of IgM is held by what type of chain? It is always J chain. Which is the least common class? Of course, IgE. Ig is involved in what? In hypersensitive reactions, in autoimmunity, in immunodeficiency. Now the answer is clearly in front of you. Answer is clearly in front of you. Then secretory immunoglobulin A is present in always in dimeric form, always in dimeric form. So these kind of very basic questions do come in all the competitions, and if you have a very clear cut understanding of the concept, it's very easy for you to answer. And this clear cut answers will increase your percentage, will increase your positive answers at compilation in any kind of competition. So with this, thank you so much for patience listening. I hope the lecture was fruitful. We'll meet again with another lecture. Till then, happy learning.